Hello and welcome to Pharma Television News Review here at the SACS conference in Basel in September 2016. On this show, I have Dr. Luc Van Hijfte, who is a CEO and founder of a company called Novithera.
Absolutely.
Welcome. Now, Novithera is an interesting company for several reasons. One is that it's actually a product of a joint venture between two other companies.
Yes.
And your focus is in the area of kinases, and we're going to talk more about that. So maybe just, Luc, if we can start, is how was Novithera formed and why was it formed?
Okay, so Novithera, as you rightfully said, has been formed by two companies. It is based on an asset that has been developed by ProKinase on the one hand, and NovAliX on the other hand.
Right.
ProKinase is a CRO, they're in Germany, and they are experts in kinases, in cancer pharmacology, in vitro, in vivo, so have really a very vast expertise in that area. And the other partner is NovAliX, which is Strasbourg-based. These are experts in biophysical technologies to understand molecular interactions. I'm talking here about X-ray crystallography, native mass spectrometry, NMR. So there we have these tools to allow then also the chemists to understand molecular interactions and how to use this information to come up with highly selective compounds.
Right.
ProKinase and NovAliX have been collaborating since a number of years on a number of kinases. And one of the targets that really struck our eye is a target called MER-TK. And so
which stands for
MER-tyrosine kinase. Absolutely. And so we embarked on that target because it's a target that has really a very high potential because it is expressed on natural killer cells and it is negatively controlled. And so that means that if one inhibits MER-TK, one would get an increased aggressivity of your natural killer cells. It means that your whole innate immune system within the cancer environment would become more efficient.
Okay.
And so at the end, Novithera, ProKinase, NovAliX, they jointly decided we will create Novithera to become also a drug discovery business, put MER-TK as an asset in there, and that was the start of the new company.
And so now you are, by various means that I understand it, coming up with different molecules that can inhibit
yes,
that particular kinase.
We have done a screen, has been done again within the two founding companies, done a screen. We have identified a number of hit series that today are binding more or less good, that still need to be optimized with respect to selectivity, but we have all the technology that we need to come up with highly selective MER-tyrosine kinases. In kinases, one of the big challenges is selectivity, especially when we tackle the ATP binding site.
Yeah.
And so we want to come up with highly selective. We can do that thanks to the technology of the biophysical technologies, and use this information to do fragment-based drug discovery, come up with highly selective molecules.
Right.
And then afterwards take these then further, optimize all the parameters that you need to become that it becomes a drug at the end.
I mean, of course, the reason why you have to be highly selective, if you're not, because there are other kinases that you can have and that causes side effects or even worse.
Yeah.
So just on that, I mean, the selection of highly selective inhibitors to MER-TK, how difficult is that to do? I mean, there's a lot of structural understanding of the structure of MER-TK, obviously the structure of your compounds,
yeah.
And so there's a lot of molecular chemistry, obviously, a lot of modeling, and so
based
forth.
on X-rays, whatsoever. How difficult is that? Again, ATP site binders of kinases. They all go towards the same ATP binding pocket that very often are quite homologous between different kinases. In the case of MER-TK, the most homologous family members are, for example, AXL and tyrosine. And so today, indeed, we are basing ourselves on the model, looking at where are the differences and try to exploit these differences based on the binding mode of our molecules, go out, reach out to differences between the pocket, or eventually either another opportunity is again based on proprietary technology that we have at NovAliX, is to go after allosteric binders, which would go and bind not in the ATP side but somewhere another side, but that also through an indirect mechanism would inhibit the enzyme. And very often these allosteric sites then, they may be more, and we would be looking at, again, at X-ray structures to understand on kind of how different between MERTK and AXL, and use again this information and come up with allosteric inhibitors. So again, the technology platform that allows us to de-risk to some extent. What we have today are series that are more ATP site binders, but again we will do fragment-based to get high selectivity, but we'll have in parallel also some approaches to come up with novel series that would tackle it from the allosteric.
So the allosteric, could you end up then with a potential combination? Because you could have two different molecules attacking the protein, and then you're more likely to come up with a very highly selective, because it's getting up to that high level of selectivity. That's the hurdle.
Does your question go towards having a molecule that binds the ATP side and another one the allosteric side? Is that your question?
Yeah.
That's a good question. I kind of need to think this through.
I'm just going to file a patent, just 1 second.
No, I'm kind of, these are already kind of having spent myself 25 years in the big pharma industry. I think it's already very difficult to go for one molecule and for one inhibitor somewhere. And I think if one inhibits the enzyme through the allosteric site in a selective manner, I think by adding then something that would be less selective wouldn't make any success.
So
on the other hand, there's, if we are talking about MERTK, which would approach cancers from, I would say, the immune activating standpoint, MERTK also has an interesting fact, is that it's involved in a number of oncogenic pathways. So MERTK by itself could also have some direct anti-tumor activity, which makes it really interesting as a target. And at the end, one of the real interesting things also is that if we really see that we have a very nice immuno-activating property within our molecules that we will develop, we can co-administer it with already known anti-cancer compounds that are out there, where then we will find synergies between kind of the direct anti-tumor killing and increasing the NK cells for cleaning up the reminders of the dying cells. So
that's more likely.
So that's more likely. And then there's another thing also, is that since last year there's literature evidence that by inhibiting MERTK, at least some of the TEM kinases, is that you get an increased T-cell infiltration within your tumor microenvironment. If that would be the case at that moment, it could become extremely interesting for some of the companies that today have their checkpoint inhibitors or PD-1, PD-L1 inhibitors like Opdivo, Keytruda. It could become very interesting to co-administer, because we really believe we would have a synergistic effect.
Yeah, absolutely. So just again, going back to the company as a joint venture, those are your shareholders effectively. So obviously research like yours requires money and financing and so forth. So how do you envisage that cash coming from?
So being at the SAX Forum here, my main mission here is to meet investors, to try to convince people on the interest of the program, which of course we are convinced of, and now it's up to us to convince the investors. So for the moment we are at a quite early stage, I should admit. We have kind of confirmed hits. There's still a way to go. The first step would be to go towards the proof of concept in vivo with our proprietary chemical series, deposit the patents and make sure that we protect the IP around what we are going to accomplish. So, and for that, to some extent, we don't need that much money. It's about 600 kilo euro that we would need for that. Then the second stage would be trying to come up with 1.6 million as a second phase, but at that moment we would already have de-risked in the sense that we have a proof of concept in vivo, interest from investors to do the full lead optimization to make really the compound into a clinical development candidate. And then afterwards go for another round of investment of about 2.5 to turn it into what we call an investigation new drug, IND, phase 1 ready. At that moment we could be already at the stage of exit, or we can take it further on and get a successful phase 1.
You have a plan, I can tell. You've got a plan. So just on that then, just a selection of one particular target as a strategy, because it's clear that you've got all the experience to do this. Company, in a sense, what's interesting about your company is that you have one specific target, yet you have a platform from which you can generate different types of molecules, get that hit up. Where in that time period that you think you will actually have a molecule that you can actually take through into IND?
The time for our leading project, to some extent I kind of was seeing kind of two different type of questions that could be behind. First of all, the timelines. Today where we stand, reasonably, I would estimate that we needed 3 years to get up to a clinical development candidate, add another year to have an IND, to have a phase 1 ready product. That means that kind of a first exit could be kind of after 4 years. Also another part of what you were saying, and I fully agree, we are here as a very young company, founded in 2016, so it's brand new. We have a first asset. It's still kind of my aim to bring this platform, which is sitting on a real rich expertise, to bring that further and to turn Novithera in the future into a kind of world leader, recognized for kinases applied to immunotherapy.
So this is your first target, basically. This is your first target.
This is the first target, and then we will start, and for the moment there's already talks with some academic institutes, also try to get maybe some kind of non-dilutive funding to fund some of this research, and start to build a portfolio of, now we have one, but in the future it should become 2, 3 different kinases that we can work upon and really become kind of recognized for the expertise, kind of exploring immunokinases.
Right. So the next stage, the important stage, obviously the science continues. The funding step is the next important stage.
It is the seed funding in the first step. When we discuss them with big VCs, I would immediately argue to get my 5 million, but at least the 600 would already allow us to take a very important step up to the next milestone, which is a proof of concept in vivo with MERTK, and at that moment would kind of bring us in the next stage. In the meantime, we would be building some other targets within the company.
Excellent. Well, Luc, thank you very much indeed for coming along on the show and tell us all about your company. Thank you.
Thank you very much. Bye.
PharmaVentures, experts in deals and alliances.